Welcome to all. I am Anitha, Assistant Professor, Research Department of Zoology, Sithalakshmi Ramaswamy College, Tiruchirappalli. In this video content, I share about the physiology of the silk gland and how to secrete the various silk proteins, their molding process of silk thread and final formation of cocoon. Let us see. Silk is produced by the insect Bombyx murri. It comes under the phylum Arthropoda, class Insecta, order Lepidopteran, genus Bombyx murri. This Bombyx murri is a monophagous insect. It feeds only the mulberry leaf. So it is otherwise called mulberry silkworm. This mulberry silkworm undergoes complete metamorphosis. It has 4 distinct stages: egg, larva, pupa and adult moth. This complete metamorphosis is otherwise called as holometabolous. The silk gland is the center of production of silk. Silk is the queen of textile. Silk gland, the silk thread of the cocoon, it is secreted by a pair of silk glands. It is a modified labial gland. This dermal gland are well differentiated in the 4th and 5th instar stage larva. It lies below the alimentary canal. It occupies most of the body cavity. It accounts 50% of the weight of the larva. The silk gland are tubular in nature and varies in different regions. First we see the layers of silk gland. Silk gland has 3 layers: outer tunica properia, middle layer is glandular layer and inner layer is tunica intima. First see the outer layer, tunica properia. It is uniform in thickness. Next layer is middle layer. It is a glandular layer. The number of gland cells in the glandular layer, it is fixed in the first instar stage larva. The number of gland cells in gland is varies in different races of the Bombyx murri larva. European race have the maximum number of gland cells. It is followed by the Japanese univolatile, Japanese bivolatile, Chinese bivolatile and Chinese multivolatile. The gland cells have the characteristic feature of branched nuclei in the mature condition. Same gland cell in young age worms, the nuclei are round in shape. Next layer is inner layer. The inner layer is tunica intima. The thickness of the inner layer is varies at various regions. The anterior region of the silk gland, the inner layer is very thick and is shed off at each mold. The other region, middle region and posterior region of the silk gland, the inner region is very thin and it does not shed off at each mold. Next is 3 distinct regions of the silk gland: posterior region, middle region and the anterior region. Posterior region is highly folded region. It lies in the midst of the dermovisceral muscle. It secretes the major protein called fibrin. It is the precursor molecule for fibrinogen. Next region is middle region. It is most prominent region of the silk gland. It folded like a W-like structure. It has 3 limbs: posterior limb, middle limb and the anterior limb. In the middle region, it acts as a reservoir of the fibrin which is secreted by the posterior region. Sericin secreted by the posterior limb of the middle region is called sericin 1 protein. It acts around the fibrin protein which is already secreted in the posterior region, which stored in the middle region of the silk gland. The middle limb secreted the sericin 2 protein. It acts around the sericin 1 protein. Next, the anterior limb secreted the sericin 3 protein. It acts around the sericin 2 protein. Next, anterior region of the silk gland. It is uniform in thickness and is very thin region. It does not secrete any material. It serves only to conduct the silk fiber which is assembled in the middle region to the spinneret region. Spindric region. The anterior region of the two sides open at the base of the medium projection in the labium called Spindric region, which drawn out the silk in the form of fine filament. The silk is molded to a thread as it passes through the silk press. It resembles a typical salivary pump. The thread of two sides are called brins. The sericine layer of the two sides bind together into a single filament called braid. The silk is a liquefied substance. When it oozes out from the spinneret, comes in contact with the air, immediately it turns into a solid braid. Silkworm oozes out the silk fluid by continuous movement of its head in a very specific manner to form the silk filament in the shape of figure 8. The larva moves its head 70 to 80 times per minute till the compact shell cocoon is formed. After cocoon is formed, it disattaches itself from the last layer of the silk of them and transforming into pupa. In the cocoon, the outer layer is floss layer. It accounts 8 to 10% weight of the cocoon. It is not reelable layer. The middle layer is very compact layer. It is a reelable layer. The inner layer is pellet layer. That is also a not reelable layer. Chemical composition of the silk. The fibrine accounts 70 to 81%, sericine 19 to 28%, remaining fats and wax are 0.5 to 1%, the coloring matter and ash containing 1 to 1.4%. After cocoon formation, the silkworm takes 48 hours. After making the shell, the larva disattaches from the silk filament and transform into pupa. Thank you.